And so this is all about some work we've really done on the Devonian-Carboniferous boundary in Greenland. And it's really led us to thinking of a new terrestrial mass extinction mechanism. And can you see a little red pointer? Anything?
Yeah.
Yes.
Yes.
Okay.
Sorry, everybody.
There we are on the top of the mountain. And you can spot the French paleontologist there with a gulwa in his hand, which is good. And everything you can see there, more or less, as far as the eye can see, is Devonian rock. Well, what this is about is mass extinction events, and what they do is they really remake the Earth. And you may be familiar with this, but it's showing you the Ordovician, Late Devonian, Perma-Triassic, Triassic, Jurassic, KT boundary, dinosaur death. And what happens is you effectively remake life on Earth every time you do one of these. And the big one at the Permo-Triassic, all of the effectively the Paleozoic animals disappeared. All the trilobites went, corals of certain sorts went, and so on. And so they're very much a part of evolutionary process, and it sort of turns over the environment. And you sort of know this story, perhaps. This is from Yehol in China, and you're looking at the Cretaceous with these feathered dinosaurs. But tucked away are the mammals hiding away nocturnally or in holes, not really noticed and waiting to effectively have their time on Earth. And these are the mass extinction events, and the nomenclature is a bit confusing in that we've got two in the Devonian, one in the late and one in the end. You can see they're not as strong as the end Permian, but it's fairly difficult to count the numbers because you've got two very close together. And it's sometimes known as the Hangenberg event. And these are the sorts of images you see, which I've obviously pulled off the internet. So that's the asteroid impact at the Cretaceous Tertiary, Cretaceous Paleogene. And there's this sort of tidal wave coming through, and everything's about to be wiped off the planet. And that's the other sort of idea, which is the sort of giant eruptions for large igneous province. And we certainly know this is true for the end Permian, Triassic, Jurassic, and almost certainly for the late Devonian, not the end Devonian. And again, you can see our furry ancestors standing there as spectators at the feast. And these are fairly passive events. This is the one on the Reykjanes Ridge on Iceland. That's Reykjavik. The lights are still burning, and then you can see the eruption in the distance. So they're passive events, although obviously planet disturbing in what they do to the environment. And that's just to show you the extent of them. That's the end Permian, large igneous province, the big one. And you can see that it's stretching about a quarter of the land area of Russia. So it's simply enormous. And if you pile it up against big eruptions, Mount St. Helens, you'll probably remember, Krakatoa, hopefully you won't. But you can see Krakatoa is a big one. But this is any large igneous province on this scale goes up 30 meters tall. So the outputs of them in cubic kilometers of lava are simply huge. And obviously we want the one on Tonga to go on this, or next to Tonga, although none of us can probably say the name of the volcano at the moment. Now, the impact of a terrestrial extinction is quite significant. And this is quite a simple figure, which works extremely well. And it's Laura Salon. And all she did was get all the literature on Devonian fish and pile it up in order of sequence. So you don't know the ages really, you just know the order in which they come. And what you can see is the world changes from a green world to a blue world and a red world. And the armoured fish, the placoderms, which are so conspicuous in the Devonian, this is Dunkleostrus about to gobble somebody up, are replaced by sharks and bony fish or actinopterygians. So all of these green things disappear, and you're basically left with a shark and bony fish world. And it all happens, you can see, all on that sort of knife edge extinction. Other things disappear. Here's the placoderms we just talked about. The ammonites very nearly all go. These funny sorts of corals, the stromatoporoids, disappear, and the trilobites almost go. So there was one or two just hang in there effectively by their tentacles, so to speak. Now, where we've worked is in East Greenland, and we're in the centre of this Old Red Sandstone Continent. So it's a rather large land area, and we're right in the middle. So it's like being in Death Valley, and we work in this rather large basin. It's about the size of Belgium. For some reason, I've got it 2/3 the size of Pennsylvania. But we're in the southern hemisphere arid zone, and everything's got effectively dry land rivers. And so if we're on the margin, we're not sure why things are happening. Here, we're recording the big events. How we get there is sort of, to a certain extent, old technology. We take a twin otter out of Iceland, and it's got these sort of fairly big tires, and that can land directly on the tundra. 70-year-old technology. This particular one I've been on many times, built in 75, and whenever I go, it seems to have changed its name and it's a different color. And here it is, basically, it's dropped us off, it's leaving, it's dropped us with the fuel, the kit, the boats, and so on. And so what we then do is we travel around the sections in a pair of Zodiac boats, and we've got to carry everything, add up all the fuel and so on. So I'll just exit out of that cursor, and hopefully so you can see about 20 kilometers there ahead. That's a nice day in East Greenland. You get nasty days, the sun's shining, and most importantly, the water's extremely flat. You do get really cold because you're traveling over this effectively a 4°C fridge all the time, and you're stationary, you're cramped up with everything around you. We meet old friends whenever we go. That's a bear. We came and effectively looked through the window, and usually we're in a hut. And one of my colleagues sort of sat up at 4 in the morning, and there was a bear about 5 centimeters from his face looking at him. And they're bear-proof windows, and all that means is the window's quite small and it can't climb in instantly, but obviously it could just push it in. That's my boot, that's a bear's boot. So you can see they are quite large. You stand no chance really. And last time I went, we had trouble. We were on one of the bases, and a bear was coming in every night into one of the expedition huts where we weren't sleeping, and it sort of bit one of my colleagues' cheeses in half. He'd obviously picked a nice French cheese to take with him. And bears have got these quite fine front teeth, and so obviously it didn't like it enough to eat the whole lot. So it clearly wasn't that hungry. It was just more curious. I mean, I certainly know my colleague would have disappeared off with this cheese in his different direction and eaten it sometime during the trip, presumably eating it from that side. There's a campsite, just to give you an idea. If I talk over this one, solar panel, and we're inside a bear fence. So that makes a bang, probably, if a bear comes in. And we've got individual sleeping tents. And there those are the boats, there's 2 boats, and they're trying to mend one of them, or mend the baseboards. And all the fuel there, well, we can only go as far as we can carry fuel, but quite a nice sandy beach to land on. This is a bit of a rougher campsite, but it's a bit windier. And again, tents inside a bear fence. And we've separated out all the cooking stuff. So every night you seal it up in barrels, so nobody can sniff anything out, foxes, bears, or whatever. And the idea is that if the bear comes anywhere, it'll come here, and then we'll hear it and be able to actually see it and persuade it to leave. One of the reasons, I'll switch cursor back. One of the reasons we come to these places are the excellent sections. So we started about here on this mountain, and in about 5 days, we got up to about there. And what we're doing, instead of drilling a borehole, we're able to take a tape measure out, get a notebook out, fairly simple technology, and go all the way up there. And you come away with a vertical section and samples from the mountain. You get very high sedimentation rates, so it really records what's happening geological. And what you've got there is what we don't like, which is a sill, and that alters the rocks, metamorphoses them. But also what it does is it drops very large boulders into the scree here, so it makes it a far heavier going as you go up the mountain. So we've worked on, there's East Greenland, that's the box, and we've worked on a number of sections I'll talk about here, probably talking about Celsius period first. But you can see it's a large area, there's 20 kilometers. And these are pollen spores. That's a very large one. You can see that with a naked eye. It's about 1/3 of a millimeter across. And we dissolve them out of rocks with this sort of nasty stuff called hydrofluoric acid. And what we can do is use these to tell the time. But as you'll see, we can do other things with them. And obviously, it's not alive anymore, but that's its resistant overcoat. And we give them names, and we can use them to effectively date rocks and, as you'll see, other things. What they're designed for is for providing ultraviolet protection against ultraviolet radiation in the atmosphere. And so you can see it's got a slight color, and you can see why. This little thing out of focus is a normal sized microspore, as is that one. So here's the Devonian-Carboniferous Boundary. And what you've got are a group of spores which become extinct. You've got a small group of survivors here, and that's what makes the vegetation in the early Carboniferous. And you've got a number of things which effectively go on holiday, all of these things. So they got to the boundary and they disappear for a while, and they've survived somewhere. And so you're going from a complicated vegetation to a simple one. And it's like winding the clock back to the early Devonian. And you can see the intensity of the mass extinction. There's the one particular distinctive spore which nearly everybody can recognize, short ranging. And here's a map of the world. There's America, there's Greenland, there's Siberia, and every dot is this spore located. So it's right through South America, right through North Africa, Australia, effectively everywhere where there's Devonian land. And so there's really hardly any plants like that today, maybe bracken, but it's enormously successful. It's got to be very tolerant environmentally, and it's probably going to function as a weed. So what it will be doing is growing fast and then just dying. And it's almost certainly what we would call homosporous, in that it doesn't have seeds or anything. It could basically make the plant again just from one spore. But despite this sort of success at colonizing the world, it just dies. This also goes, and this is the scruffy vegetation. That's the understory vegetation spore. So we lose that one completely. And the forest tree gets enormously disrupted and we lose most of it for quite a while. So what you can see is that you're hacking at the keystone elements in the ecosystem. So it's not surprising that fish struggle because the environment in which they lived was basically being ripped up. Now, we wanted to match up the spore and pollen records with the forest trees. So these are some of the forest trees. And we're off this mountain called Celsius Berg. And there's the Devonian-Carboniferous Boundary up there. So all the interest was up the top. And normally it'll take you about 4 hours to get up. And as your knees get older, it takes you 4 hours to get down again. So we sort of cracked the secret in what we would do was go and I have to change cursor to do this. We cracked the secret and we basically just lived on the top at 1,200 meters. Oh, no. Backwards. And you can see where we were working. That's the Devonian-Carboniferous boundary there. And if you look in there, there's a quite a little figure just there. And so you got it all across the screens. So quite simply, all we did was the three of us stood in a line and we went up, putting it with altimeter GPS, got the tape measure out, and we measured the width of the plants. We just counted up the numbers. And what you can see, here's the extinction of the Devonian forests, happens at exactly that point. Here's a section on another mountain, Stensioberg. We found it in 1996. And if you ever do microfossils, the problem is that you can't see them when you're there. So you don't know that you stood on the Devonian-Carboniferous boundary until you get home. And so it took till 2006 to get back to it, and we got back in on 2006, 2009. And it looks a bit of a mess, but you can basically dig it out and you can get some quite decent samples out of it, and you can sample it at 10 centimeters or so resolution. And this is what you see. So there's the Devonian-Carboniferous boundary, and there's two lakes, one lake and two lakes. You're in a terrestrial environment, and it almost dries out, but not quite. And you can see all these Devonian plants all become extinct, disappear at that level. And the lake's got an enormous carbon content in. So that's 20% carbon. That's simply huge. And so if you pick up these rocks, they actually feel quite light. But the problem is that you don't see what's happening in here because of all the organic matter effectively crowds everything else out. So what you have is Devonian slime, and that just masks everything. So the real question was, how did this extinction, did the things tick out slowly? Did it all happen, one afternoon on a Thursday? Now, we'd always known about this section of a place called Rebel Backer, and we had a couple of goes. We dropped in sort of on it while boating, 2012, and worked on the west side where it was accessible from the coast. And the thing about it was we're predicting that would be on the edge of a lake. So we should see much better record of pollen and spores through it. And we knew the boundary was in there because the Greenland Survey had gone up there in 1985. And what they were doing was being dropped at the bottom of the mountain in the morning, and they would then hack up it and be picked up by the helicopter in the evening or past of the evening. And so they would do that day after day after day on different mountains. So they weren't quite sure where it was, and we weren't quite asking them properly. Well, we were and we weren't where it was, so we couldn't find it, and they couldn't tell us where it was. That's interesting. And I have frozen on the screen. I've got a whirly wheel at the moment, and I've got a frozen cursor. What I'll do is I'll try. Oh, it says, can you see that? Microsoft PowerPoint's not responding. I've not had that one before. Can you hear me at the other end?
Yeah, we can hear you.
Yeah, fine. Everything just stopped. Aha, right. I think we're back.
Right.
Perfect. Good. So, you know, when you're flying over these places, you take pictures. So that's a random overflight. I didn't really know what it was, and I took a picture of it, and that gave us a view of the other side of the mountain, much better than anything on Google Earth. And I was able to see those potential sections. So in 2017, I took a chance on a helicopter. So we were 12 kilometers inland, and so we just basically devised this quite short trip, and just got dropped directly on the spot, maybe. So that's the helicopter leaving us, and that's the point at which you hope you got everything. And these are all the ridges you could see on that cabin window picture. And effectively, we decided to go there, and the section was here. So we were quite lucky. 7 ravines on that side. And if we'd come over the ridge in, say, 2012, we'd almost certainly have gone into the wrong ravine where the boundary wasn't. And at that point, we'd probably have given up. So I went to Ian Troth, who's been mentioned, a former student who was did a PhD in Bolivia. And again, it's quite simple. We just got a tape measure out and off we went. And we're looking for 0.5 meter of mudstone in that section. And that's sampling it through, and you don't know if it's the boundary again until you get home. It's not a very good picture because you're clinging to the side of this ridge here, so you can't in fact actually step backwards and take a good picture. There's the sort of what we got out of it, and you could see there's a fairly swift extinction of everything. That's one of the boundary one, this Lepidophyta, which disappears. And so you could see it was a knife edge extinction at that point. Everything Devonian went. But the big surprise was the malformation. And there's a happy normal spore, and it's got regularly spaced spines, and they're all got pointy tips, and they're all the same length. But what you found was most of them weren't like this. Most of them were like this one. And what they've got are tips which are malformed, the spacing's gone wrong, and they're basically merging in. This was a good one. Here's one where the spacing's gone and they've sort of gone to clubs. Here's one where they've just twisted up and the spines are all connecting in a sort of mesh. It's always the same species. You can see it's got 3 spots, but there it's nice regular spines. It knows it's got to grow them, but it's lost the ability to do it. And this one is sort of like effectively a worse shape, and it's just dumping spore walls, spore spine material in approximate places. And this one's given up trying to grow the outer coat. That's a tetrad of 4, and you can see the little spots in the middle. And that's one, a lot of them do this, in that they're not dropping out of their tetrad in how they initially form. So they're not splitting from meiosis. And the other thing you can see, it's dark, it's pigmented. So this thing is sterile, it's never going to grow into a plant. Now, here's some spores, and they've all got quite regular patterns of how they grow those spines. And it's under gene control. And so what the little cell in here is doing effectively is saying grow spines, this length, that tip. It's like us growing fingers or hair or whatever. It's telling it what to do exactly. And what is its problem is that it's had a blast of the ultraviolet light. Now, there's a spore mother cell, splits into 4. Because it's splitting into 4, it can only then put its protective coat on after it's done that. So there's a point at which the plant is holding it towards the sun, saying, I dare you to irradiate me. And if it gets irradiated and it gets UVB damage at this point, it'll effectively damage the DNA. And that's what then makes it unable to form the right sculpture. And what happens is adjacent base pairs covalently bond. So when the DNA tries to unzip, it basically comes to a stop in the zip. Now, plants are clever things. And if you put a pot plant under ultraviolet light, what it will do is uptick the UV. So it's no difference to some of us having the ability to tan. And so a plant will do the same thing, and it will put UV absorbing compounds into that wall. I mean, probably most people in Cumbria are like me and turn red. So here we've got the bottom of the lake. Here's the proportion of malformation. You can see it's running at 80% in that species. And what you can see is as you head into the center of the lake, the pigmentation gets stronger and stronger. So I've measured translucency of those walls. And so you can see what we can really show is that we're getting ultraviolet damage. I mean, that's why pollen's yellow, because it's giving it some protection. But by going browner, you're giving it more protection. And it's not the only spore which does it. There's another one, nice sculpture, nice color, into the boundary layer, and it loses the ability to grow properly. It takes pigmentation on, and you go above, and it's effectively gone back to what it was. Now, this is sort of known. So when you get large igneous provinces, Permo-Triassic boundary, you get a normal bisaccate pollen, and this one is malformed. And so it's not got 2 sacs, it's got 3 sacs, it's got 4 sacs. It's in twins which won't split. And the other signal we can get for a large igneous province damaging the ozone is there's the volcano, and that produces mercury. And that's where mercury comes from in the atmosphere, just volcanoes. And so the idea is that if you can measure the mercury, you can see if you've got a large igneous province. Mercury is captured on organic matter, it loves it. And so what we do is we divide our mercury content by our carbon content, and that will show us an anomaly. So there's a volcano pushing out mercury. Mercury is dropping out, stays in the atmosphere for less than a year, and then it gets drawn down into the organic matter and so on. So here we've got 3 sections in East Greenland, in the same lake at the Devonian-Carboniferous boundary, and there's lots of mercury, 200 parts per billion. But once you divide it by the carbon content, you've got no mercury. Here we've got another section, you're running up to 10 parts per billion, nothing. And then if we go along, same lake, same level, into the lake edge where we had a malformation, you've got hardly any mercury, a few parts per billion, hardly any carbon, and it's flatlining again. So what you can say is that on this evidence, there is no reason for a large igneous province. Now, I've taken you deliberately here to the Newark Rift in America, and that's the Triassic-Jurassic boundary about, we're below it. But what you've got is a lake cycle, and there's the permanent deep lake, dark coloured mudstones. That's the Playa Lake, sort of salty. And here you've got the lake edge, and you find dinosaur footprints in there. And so what you've got is the same process acting all these lakes in East Greenland, Triassic, Jurassic, and also lakes which you may have come across, and there's lots of concern about Lake Chad at the moment. So here we've got the modern Lake Chad, and it's tiny. And it's drying out, and it's drying out because that's what it does, because it's on this position between the desert and the equatorial, effectively the rain coming over the watershed. Now, if you go back 10,000 years, what you find is there's much more heat in the atmosphere, and that's causing a stronger monsoon. The monsoon in East Africa is sufficient to fill every single one of these Rift Valley lakes, and then discharge into the Indian Ocean. It also fills up Lake Chad to be mega. And so you can see it's highly environmentally sensitive. And all of that you can see in this one figure from Arabia. So there you've got some old graffiti, several thousand years old, and these are probably people. That's probably a shaman, and that's an ibex there. And so they're off hunting it. And that's the time when it was wet. This is the time now when it's arid, and so the more modern graffiti is a camel drawn over the top. So what you really want to know in your Devonian world is what's on at the South Pole. So we know what's happening in the sort of the arid zone, the Southern Hemisphere arid zone. And so there's Bolivia there, Devonian South Pole is around here. So in parallel with some of this work, we had a student who've seen on the boat and eating his tea, John Lakin. And that's effectively his field area. That's Lake Titicaca. We're up at about 4.5 kilometers. And we're looking at these glacial sediments, or the end of the glacial sediments, at exactly the Devonian-Carboniferous boundary. So it's quite hard up here because you've got to effectively climb up from the road. So you're sort of moving in slow motion and not wheezing. And so what is the big thing from his work, what we showed was that if you measured section after section through the boundary, what you got was a channel. And that's it drawn out as sections. Here it is drawn out at vertical scale, but we've just got half of this channel. And so that's down cut by about 100 meters. And it's filled with glacial sediments, or sort of end of glaciation sediments. So that's effectively the ending of the last terminal Devonian glaciation. Now, Devonian glacial sediments are known in other places. Here's Greenland, and they're known down here in sort of Pennsylvania, Maryland, and so on. And what you'll notice is that's closer to the Devonian equator than the last glacial sediments got to the modern equator at sea level. And so it's a more intense glaciation. This is a close-up. There you've got Pennsylvania, Maryland. And the other thing to notice in the outcrop belt are these boulders here. And we'll take you to see the Robinson boulder. But what you have are diamictites and glacial sediments, and everybody was happy it was like Mount Kilimanjaro and are effectively coming off highlands, so you could trade latitude for altitude. But then this one turned up, the Robinson boulder, and it's a loamstone, and they'd long been known around, but always were thought to be quaternary, despite being south of the ice limit. But this one was encased in top Devonian mudstone. So that's a genuine dropstone. And that means that you've got tidewater glaciers at about 35 degrees paleo south. So it's really quite significant. So we're able to put this together. Here's our Greenland section. Here's Bolivia. There's our termination of the glaciation with a 100-meter down cut of sea level. Here's our Greenland section, and that's our aridity of the glaciation. And what we looked at with the mass extinction was the warm up here. So what you can see is extinction is an extremely warm lake associated with the coldest, most close to the equator glacial cycle. And this is Germany, which gives you the effectively the standard time. And so we then came up with a model which looked at the effects of warming on glacial or warming on continental areas. So there's data here from the mid-continent of the United States. And when you get very hot summers, you get moisture vapor going up higher, and these are naturally occurring ozone destroying fluorocarbons, which I've drawn. And those are produced by microbes, often marine microbes, salt marsh microbes, which might encounter sodium chloride. And so into their metabolism, they effectively spit out a naturally occurring fluorocarbon. And that gets pushed up higher towards the ozone, and then that starts to thin it. You increase the UVB, and you start to destroy the reproductive ability of the forests, which collapse. That flushes nutrients into the oceans, at which point you generate more fluorocarbons and they go up. And you get out of the cycle because you're in a Milankovitch-driven climate cycle of a short, sharp glaciation and then an equally short, sharp warming up. And then it'll turn the corner of the next Milankovitch cycle and naturally cool down. And so that was, you know, all quite exciting. And the message would be that one of the problems with global warming is we might start to thin the ozone layer. Now, what then happens is, of course, the mass extinctions. When you write a paper on mass extinctions, you get lots of emails from lots of people. And the astronomers became involved because they've always had this assertion that, you know, they were successful and they told us the KT boundary was an asteroid. And they then said, well, what we've always told you, you've never listened, is that a supernova blast from a star collapsing will thin the ozone layer down. And that's really the cause of your mass extinction. At which point they obviously then interpret all mass extinctions on Earth from supernova. Now, the one thing they've usefully done is they've highlighted that in the supernova collapse process, you get certain chemical elements made which you can never find on Earth, apart from probably around Cumbria. And that's plutonium-244. And this is effectively made in the center of these supernova. Now, the supernova blast happens, and that's the speed of light, but following it at a distance is a dust cloud. And the idea is that we can then test out this supernova hypothesis once and for all by looking for plutonium. Now, this is a positive upside from Chernobyl, because there's been a lot of research on plutonium-244 and measuring it in the natural environment. And in Southampton, we've got a environmental radiochemistry group, as in Lancaster. And you can see why Lancaster's got one. And what the UK government effectively is investing in is one of these, which is an accelerated mass spectrometer. And what this could do is measure plutonium-244 and its friendly other isotopes to the 10 to the minus 15 gram level, to the femtogram level. And so the next part of what we want to try and do is I'm going back to Greenland this summer with the tetrapod workers looking for fish with feet, and bought a backpack drill, which is that. It's basically a strimmer with the handles on the side and a water pump, and that can go down and sample several meters depth. And the idea is that we're going to go back and collect some pristine good samples for plutonium analysis, and we can test this supernova hypothesis. And this, I think, is about the end. So if I get into there, you can see us going home. End of season, it's nearly September. We go in August because the ice is thinnest then. So that's 2 expeditions going back together. So that's on Ella Island here. And that's interesting. That cliff is a straight 1,200 meters drop to the bottom. So you quite happily go and stand on that, but it's quite scary. And that should be it. We thank Cambridge Arctic Shelf Programme, now CAS, for logistics, getting us there, getting us back, and National Geographic for important funding. So there we are, about to go home in a different trip. Thank you. So if I pass control back. And that's it.
Thanks, John. Right. Well, I'm sure, having heard a talk that touched on so many different topics, there's going to be questions asked by the audience. So I'd like to invite anyone who wishes to pose a question to John to do so now.
I see Clive's not going to ask anything. Oh, he is.
Yes, Stuart.
Hi, John. Good to see you again.
Yes.
And I was fascinated by these pictures of the deformed spores. Is the deformation itself caused by the ultraviolet radiation? I mean, one of the things that came across is these looked remarkably like COVIDs, you know, at the present time. But is it a radiation-induced deformation that's taking place?
Yes. Well, yes. Botanists work on this with crop plants. And so they will irradiate crop plants with UVB and they can get similar malformation. Now, what they've never done, obviously, is done it to something like bracken or a spiny living spore, such as we see alive today.
Mm-hmm.
And so those are some experiments we're hoping to do if we get some money, etc., is to run that sort of thing. But yes, it basically gets the DNA.
Yeah.
Because
Are
what
there
you're doing
modern
is-
analogies of the sort of processes that you're observing at the Carboniferous Devonian boundary?
what are the
Modern analogues.
Oh, well, yes and no. No, I mean, you can experiment it, but what we possibly think is that, you know, we think Cambrian, Precambrian, whatever, ozone layer forms and it stayed there.
Mhm.
Because we've got high sedimentation rates, we're able to see quite well what's happening on a very fine time scale. So it's maybe that there's other times in Earth history the ozone layer has quietly thinned or departed.
Right.
And so now the challenge is to actually measure it through geological time. And it's probably something like a seasonal thinning. So it'll vary depending on where you are in the world, and it'll depend what sort of plant you are. So if you've got your reproductive bits up to the sunshine,
Yes.
which is where you want them to throw the stuff on the ground, that's the worst possible place.
Yeah.
If you're a seed plant and you've got them inside something, you're probably in a better state.
Right. Right.
A lot of plants, probably, you know, the survivors probably grow vegetatively, or they're long lived. In other words, if you live for 30, 40, 50 years, you only need to reproduce once. If you're an annual, like that one which suddenly went extinct despite being global, you know, you just need 2 years and you're gone.
Yes.
You're wiped. That's it. So all sorts of questions are sort of asked by this.
I'm sure. Yeah. Thanks very much, John.
Thank you.
Hi, John.
Hello, Clive.
Question about tests of hypotheses.
Yeah.
More tests for your hypothesis, the sort of natural ozone killer, and for the astrophysical approach, any more tests there that can be done besides looking for exotic isotopes?
Yes, we can test the what you can do is you can use Fourier transform infrared analysis on a microscope, and they're good enough to point at a spore, and then you can actually recognize the absorbing compounds inside the wall. And so what we should be able to do is go through a sequence of these spores through the lake, and we should predict that we'll find more absorbing compounds in the walls are the ones which are malformed. So that'll be the test of that. The isotopes which you get, there's quite a few which you get from a supernova, but most of them have got half-lives which are too short. So it's plutonium's the exotic, uranium is long enough, but there's too much of it on Earth. Now, the good thing about plutonium 244 is that there's 240, 242, which are also made in bombs and in supernova, but they've got very short half-lives. So if we get 244, we can check it that we've not got any contamination from a natural environment and on site. The interesting upside will be because the UV blast comes at the speed of light, the dust comes somewhat slower. If we can measure the distance between the two, because we're in a laminated lake, we know the seasonality is annual, and we should be able to estimate in years the time distance between the arrival of the flash and the arrival of the dust. And that will tell us how far away the supernova is. And if we can find an astronomer with a big enough brain, we can then get them to work back to what likely supernova it was which did this, or where it was coming from. We think it's got a double upside, because if we find plutonium 244 in there in enough quantity, you know, we've proved the astronomers right and fair dues. But that's a really good discovery. if we can't find it, we think they're wrong. They'll say, of course, it was something wrong. It's a different supernova type or something. And so we can basically tell them to be quiet for a while and look at other hypotheses. So it's maybe a double win.
Indeed.
I had to learn what a femtogram was, which is 10 to the minus 15 grams. And I had to write it out once in full, which is 15 zeros.
And Crowder can measure that, can he?
They claim so, yes. On this new AMS system, they claim better than 10 to the minus 15. And they can more or less count them as they come off. But this is why we want, big samples. And the chemistry you have to do is fairly straightforward. But I mean, I don't worry about that. It'll be their problem, not mine.
So what size of sample do you have to collect, John? You say a big sample. I'm just thinking your logistics.
It's absorbed in organic matter. And so it's really just grams, but we'll probably take a 10 gram split for a core of a whole of that 10 gram to try and capture it in case it's in 2 centimeters of that 10 gram. Then homogenize 10 grams and then it goes through an acid leach. So it's quite straightforward. And we've got everything going for us because it's an organic rich lake, which likes locking onto plutonium. And it's also a very big lake. So it's got a very big catchment area. So it's got a lot of rainfall and it's got the ability to capture that, obviously, the plutonium as it comes in as dust. So you're concentrating it in the big catchment. You're then concentrating it with a lot of organic matter. I mean, you have to go try these things in hope. If it wins, it wins. If it doesn't, but it's funded by people looking for fish with feet, tetrapods. So they've got ability to get money for other stuff, and we're going to do something quite different. But obviously, we're going to drill the holes where we're there.
So are you obliged to collect tetrapods for them? The sideline? Well,
yes, I'm going with them, and my job there is to do time correlation and sort of basically do basic palynology and so on, and to try and understand the environment of this paleo lake. So they're interested in me cutting cores for it for other reasons, because the lake has tetrapods in. So. And obviously, I've shared with them that this is what we're going to do, so they're very keen on this.
Yeah,
because it explains why their extinctions happen. So they're very positive about it.
Yeah. But does anybody else have a question for John? Either they've all gone away and just left themselves logged on. Or you've just stunned them with such an amazing talk, John.
Yeah, don't worry, because there's a lot of different things in it. Anyway, I'll give way to Hugh Clough, and I think I saw Fiona Hyde